Neuromuscular diseases are degenerative and progressive disorders, impairing signal transmission between motor neurons and muscles. They can occur in childhood or adulthood, and their most frequent symptoms are muscle weakness, stiffness, twitching, torpidity, cramps, myalgia, and loss of muscle control. They can directly impair muscle function or nerve function. This in turn impairs muscle function. In most neuromuscular diseases, genetic mutations have been found. They affect about 500 genes, so generating a wide range of heterogeneous clinical conditions. Mitochondrial dysfunction plays a key role in their pathogenesis. Mitochondria play an essential role in cell metabolism and energy production through the oxidative phosphorylation pathway. Notably, in nerves and muscles, we have a high density of mitochondria because these tissues are highly dependent on oxidative phosphorylation for their metabolic needs, so they are very sensitive to mitochondrial dysfunction. Furthermore, mitochondria are implicated in calcium homeostasis, apoptosis, and ROS generation. A normal ROS level is involved in homeostatic signaling pathways, cell differentiation, and cell proliferation. But an increased ROS level causes mitochondrial dysfunction, as found in skeletal muscles of the affected patients. Genetic mutations have been found on genes, on mitochondrial or on nuclear DNA. These genes may encode subunits of the respiratory chain complexes, mitochondrial enzymes, or mitochondrial carriers. As regards mutations found in the components of the respiratory chain, mutations have been found in core subunit of complex 1 and also in subunits belonging to the N, Q, and P module of this complex, so affecting its function. For complex 2, mutations have been found in the gene encoding succinate dehydrogenase subunit A and also in a gene encoding an assembly factor. In complex 3, mutations have been found in the gene encoding cytochrome B and also in some genes encoding subunits with a structural or a catalytic role, and even in genes encoding some assembly factors required for the proper folding of this complex. In complex 4, mutations have been found in genes encoding core subunits of this complex and also in gene encoding assembly factors. In complex 5, mutations have been found in ATP6 and ATP8 genes, which encode subunits A and 8 of the F0 domain, respectively. They are involved in proton transport and assembly stability of this complex. So this mutation can affect ATP production linked to proton transport. In general, mutations affecting respiratory chain leads to a decreased ATP production with increased loss of electrons through the respiratory chain. This results in increased ROS production, especially in complex 1 and complex 3, which are the main ROS producers. In this slide, you can see mitochondrial metabolic pathways involved in the pathogenesis of neuromuscular diseases. Optic atrophy 1 protein is a GTPase responsible for the fusion of inner mitochondrial membrane and mitochondrial DNA maintenance. Its mutation results in altered mitochondrial morphology and function with mitochondrial DNA deletions. Mitofuscin are also GTPases involved in the fusion of the external mitochondrial membranes. Mutations found in the gene encoding mitofusin 2 are responsible for sensory and motor neuropathies. Also, the genes encoding some enzymes of the Krebs cycle can be mutated, such as aconitase, isocitrate dehydrogenase, succinate dehydrogenase, and malate dehydrogenase. In these cases, we have a decreased energy production with increased ROS generation, with important pathological implications. The carnitine palmitoane transferase 2 is an enzyme involved in the activation step of fatty acids before their beta oxidation. Mutations found in this gene are responsible for carnitine palmitoane transferase 2 deficiency with different associated phenotypes, which depends on the residual activity of this protein. This affects beta oxidation with decreased energy production. The same consequences are seen when mutations are found in the gene encoding acyl-CoA dehydrogenase, in particular short-chain acyl-CoA dehydrogenase and very long-chain acyl-CoA dehydrogenase. Other mutations have been found in the gene encoding electron transfer flavoprotein and its dehydrogenase. Normally, these proteins are involved in electron transfer from beta oxidation to the respiratory chain. Their impairment results in multiple acyl-CoA dehydrogenase deficiency with severe neurological and muscle symptoms. Also, the gene encoding aryl-CoA hydratase can be mutated, leading to severe phenotypes. Here you can find the neuromuscular phenotypes related to the alterations we have already seen. Note that in the case of isocitrate dehydrogenase, mutations in this gene are not associated with neuromuscular symptoms in humans, but only to retinal degeneration. On the contrary, in Drosophila melanogaster, mutations found in the corresponding gene are associated with impaired synaptic transmission. Here you can find the neuromuscular phenotypes associated with alteration of lipid metabolism. Here you can find neurological symptoms, muscular symptoms, and also cardiac symptoms. This is because the heart uses mainly fatty acids for its energy production. Furthermore, lipid accumulations due to defective beta oxidation results in impaired mitochondrial function with decreased energy production and increased ROS generation. Also, the genes encoding some mitochondrial carriers can be mutated. These are transporters located on the inner mitochondrial membrane. They belong to the Slc-25f family, the largest solute carrier family found in humans. They transport metabolites across the inner mitochondrial membrane, so providing a link between cytosolic and mitochondrial reaction. They have an essential role in cell metabolism. Here you can find the citrate carrier. It translocates mitochondrial citrate in exchange for cytosolic malate. Its role is essential in biosynthesis of fatty acids and sterols, as well as in gluconeogenesis and other important metabolic pathways. When mutations affect its activity, we have congenital myasthenic syndromes with different severity. It depends on the degree of residual activity of the carrier. When we have the total loss of its activity, we have a decreased flux of intermediates through the Krebs cycle with impaired energy production and increased oxidative stress. Here you can see the phosphate carrier, which transports inorganic phosphate in the mitochondrial matrix. Alternative splicing of its gene originates two different isoforms. Isoform A is mainly expressed in the heart and skeletal muscles, so its impairment implies cardiac and skeletal muscle symptoms. In the case of isoform B, it is ubiquitously expressed but at much lower extent. The most severe phenotype is associated with mutations which affect the activity of both isoforms, with shorter lifespan. Here is the Adp Atp Carrier, which translocates cytosolic ADP in exchange for mitochondrial ATP. So this carrier is involved in ATP production and balance of nucleotide pools. Mutations affecting the activity of the isoform 1 of this carrier are implicated in 4 different associated phenotypes. The severity of the symptoms is related to the residual activity of the carrier and also to the type of defects involving mitochondrial DNA. In particular, the most severe phenotype is seen when we have a loss of activity of this protein associated with mitochondrial DNA depletions. Here you can see the aspartate glutamate carrier, which catalyzes the electrogenic exchange of cytosolic glutamate for mitochondrial aspartate. This is a key component of the malate aspartate shuttle, which transfers NADH reducing equivalents from cytosol to the mitochondrial matrix, so taking part in ATP synthesis. Also in this case, mutations can affect its gene, in particular the gene encoding isoform 1. In this case, we have neurological, muscle, and cardiac symptoms. We have also hypomyelinations because the low level of aspartate present in the cytosol impairs myelin biosynthesis. Next, we can see the carnitine acylcarnitine transporter, which catalyzes the exchange of cytosolic acylcarnitine for mitochondrial carnitine. Its activity is linked to beta oxidation, and also in this case we have mutations that can interfere with its activity. We have 2 different isoforms. The severity of the symptoms is always related to the degree of residual activity of this protein. Here we have the 2-oxo-dicarboxylate transporter, which catalyzes the mitochondrial import of 2-oxo or 2-amino adipate, which derives from the cytosolic degradation of lysine and tryptophan. Mutations on the gene encoding this carrier cause spinal motor atrophy-like disease. In these cases, the accumulation of 2-oxo adipate and crinolinic acid in the cytosol is responsible for neuronal toxicity. Here is the folate transporter, which is believed to transport flavin adenine dinucleotide and folate in the inner mitochondrial membrane towards the mitochondrial matrix. so its role is essential in the regulation of one-carbon metabolism and also in redox balance. Mutations found in this gene are responsible for two different associated phenotypes. In this case, we have impaired ATP production because we have low levels of fat in the mitochondrial matrix, which impair the function of mitochondrial respiratory chain flavoproteins. Then we have the coenzyme A carrier, which transports coenzyme A in the mitochondrial matrix in exchange for adenosine diphosphate. It has an essential role in many metabolic processes, including the urea and the Krebs cycle, beta oxidation, EMA biosynthesis, and so on. Also in this case, we have mutations that can impair its activity, leading to severe phenotypes. Lastly, we have the Slc-25f46 member. This is not believed to be a mitochondrial carrier. Rather, it seems to be involved in mitochondrial dynamics and fission. It is located on the external mitochondrial membrane, and in vitro experiments have shown its involvement in mitochondrial fission, as you can read in the slide. Here you can also see the associated neuromuscular phenotypes. In conclusions, we can say that mitochondrial dysfunction plays an essential role in the onset and progression of neuromuscular diseases. Given the lack of approved therapy to treat these diseases, it's essential to understand the link between them and impaired mitochondrial metabolism in order to develop new therapeutic approaches to treat these pathologies and to improve quality of life of patients. If you are interested in this topic, you can contribute through this special issue for which I serve as a guest editor. Thanks for your attention.